because there is unrecognized diversity of reptiles on islands, so they make a good system to study. So, why are reptiles so diverse on islands? It probably has something to do with their life history traits, so they can raft really well. So this is actually not an island, but a raft that was off South America a couple of years ago. And this, you can imagine that there's a whole ecosystem going on this raft, and that if that raft were out there long enough, eventually the mammals and the birds on it and the frogs would probably not make it for a long period of time across the sea, but reptiles can do it. And they can do it because, of course, they have a low-ish metabolism. They don't need a lot of food. If they are struggling to find food, they can go without. And the same with water, so they don't need water every day. They can hydrate pretty easily by just licking a little bit of dew off of leaves and things like that. And they're real survivors, so they can undergo these long journeys on rafts, certainly better than mammals and frogs, and probably even better than birds. They can do it as adults, and they can also do it as eggs. So a lot of reptiles can just delay their, the eggs can delay their development and hang on for a while. So good on rafts, but some reptiles don't even need rafts. So this Aldabra tortoise washed up at one point in Tanzania. It's a 700-kilometer plus journey, and it did it without a raft, and they reckon that it was at sea for more than a month by the size of the barnacles on its legs. So reptiles, I would say, are really good dispersers across the ocean. And one of the more famous examples, obviously, is the Galapagos. So all kinds of organisms can get to the Galapagos pretty easily because they've got lots of time to do it, at least 4 million years they've had, and it's a long journey, but it's not unheard of to get there, obviously. 1,000 kilometers on a raft, you can make it. And the currents are favorable for that. So things can get there. And reptiles certainly, reptiles can get there probably better than other things. So there's 38 native reptiles-ish. I actually struggle to find good species lists for most of the islands. So there's already a research gap. We need really good lists of what's out there. But reptiles, about 38 probably on the Galapagos. Of course, some of those would have speciated in situ, but there's multiple colonization events. There has to be, because there's at least 4 different groups of reptiles on the Galapagos, and of those, many of them have multiple genera, so it has to be separate rafting events. So reptiles getting there really well. If we look at the area where we are now, the mascarines, well, this is a little bit more tricky to get to than the Galapagos. So, I'm going crazy with this. So the nearest landmass is Madagascar, and Madagascar has plenty of reptiles to raft over to the mascarines. That's absolutely true, and there are some reptiles here that are relatives of Malagasy reptiles. So it has happened. We have about 16 on Mauritius, 16 reptiles, of which 5 have already gone extinct due to human impacts. There are 7 reptiles that only occur on the little the little islets around the island, so they've gone locally extinct on the island itself. And there's some introduced species. On Reunion, there were at least 7 species here, but now there are only 2 because 5 have gone locally extinct, or extinct on the island as well due to humans. And there's a whole load of introduced species. You've probably seen them running around outside already. So 90% of the reptile fauna on Reunion is introduced. So it's a big number. But of the native reptiles, some have come from Madagascar, but some have not. So where did they come from? So here's my pitch about reptiles being really good dispersers. So this is the ocean current, and we need to play the video. There it goes. So the prevailing current obviously is toward the west, with currents hitting Madagascar and then sort of making a little back eddy toward the mascarene. So stuff from Madagascar can get here in those back eddies. But most of the current comes from this direction, and that's a massive, it's the massive Indian Ocean to get across, and reptiles have actually done this. So it's a minimum 6,000 kilometer journey from the nearest landmass, can be even longer. And there is at least one skink that has done this. It's on Mauritius. And this is a phylogeny that includes that skink. and its sister group is found in New Caledonia. New Caledonia, obviously on the other side of Australia even, so it's related to things far distant, probably 7,000 kilometers away. And this isn't a once-off either. So there's also a gecko native to Mauritius, and it hasn't been included in any phylogeny, but it's in a genus that occurs also on this side. So it would have also had to make a huge rafting trip to get to this where we are now. So that's my pitch about how reptiles are really actually quite a diverse group on islands. They've gotten to islands, they can do it. And I'm going to actually leave the rest of the important pitch about reptile diversity to the experts. There's 4 herpetologists and a geologist, and they are going to talk about the patterns of diversity on islands, the evolutionary mechanisms, and the sort of speciation and colonization. So with that, I'm going to call the first speaker, which is Luis Ceríaco, who's from Porto, the Natural History Museum in Porto, and he will be telling us about the Gulf of Guinea island reptiles.
Okay, this is working. Thank you, Crystal. Thank you all for being in here. As Crystal told, I'm going to talk about the Gulf of Guinea, which I've been working there for a few years so far. And I would like to reinforce what Crystal just told us about our knowledge about reptiles on islands, because as I'll show you on a little bit of history of the knowledge we have on the Gulf of Guinea, which is a fairly easily accessible place, the things that we have been discovering a lot of stuff in the recent years. So where is the Gulf of Guinea, and what are the Oceanic islands in the Gulf of Guinea? So they are situated in the west coast of Africa. And they are constituted by 3 main islands that are really, really old. They are part of the Cameroon volcanic line, and they start here with Principe, that is the 31 million years old, followed by São Tomé, then Annobon. Bioko, we not consider it like a truly oceanic island because it has been connected to the continent several times. So in my talk, I'm just going to focus about these 3, especially the first 2, which are the ones I've been working more. Due to the old history of these islands, but also because where they are situated, the islands are quite homogeneous in terms of its landscape. It's like a West African landscape, typical West African landscape. But of course, there is some already changes due to human uses of the land. So these islands were colonized by the Portuguese when they got there in the late 15th century. And of course, people have been there since then. And there was a lot of agricultural uses, first with sugarcane, then cocoa and coffee. And now some areas have some use of palm oil, etc. Also some islets around it that I'm going to talk also. Of course, are not as luxuriant as the main islands, are like more these rocky landscapes without any plants. But also due to this old geological history and this habitat and this placement near the West Africa, really diverse area, these islands are rich with biodiversity and with some of the biggest endemic percentage in the world for birds. There's one of the biggest endemic percentage in the world for an island. For example, amphibians, there's a lot of amphibians that occur in here, and they're all endemic, including the Sicilian. There's 2 endemic land mammals, shrews, and reptiles. I'll show you there's also really interesting diversity. And if we talk about plants or insects, it's even bigger, and we even have less knowledge. So one of the big questions, and especially for some animals that are poor rafters, like amphibians and mammals, is how they get here. And the interesting thing is that the ocean currents help a lot in this situation, because the islands are situated here, and besides we're having a lot of big rivers that flow directly to this area, like the Niger and the Congo, there are also a lot of big and important currents that push things towards there. The Bengala current that pushes things from the south up to São Tomé and Annobon, and all the Guinea current that just like pushes things that come from the Niger and just like towards Principe. So this, like, there's a lot of currents that help things going there. And also it's not that far from the mainland. There's been some papers about how things got here, and there's an important paper by Misi et al. About how the amphibians got there, because it's quite difficult to explain, and especially having like animals like caecilians in here. And it has been proposed that the combination of the currents plus like a change in salinity at certain times of the year can help these animals to survive the movement from the mainland here. What is interesting also is that when we look to the biogeographic patterns of these islands, they are not equal for island to island, and there are patterns that are repeated in all islands, others are just specific for one another. And while in general most of things are similar to what we have in West Africa, there are some other things that are more related to things in East Africa or Southern Africa. And part of this is explained by the currents. So let's just talk a little bit about the history of herpetological works in here. So the first records we have about reptiles on these islands are from the Portuguese sailors that got to there. As I told you, the islands were discovered by the Portuguese in the late 15th century, and in the early 16th century there was this interesting report about a Portuguese navigator that places some animals, the reptiles there, namely crocodilians that are no longer there, and so they were extirpated by the colonists, and also a venomous black snake that I'm going to talk a little bit further, which is a Naja cobra. So after that, we waited a lot of time for having new reports for reptiles on these islands, and in the early 19th century, there was several authors that work with stuff from there, because the boats that came from Africa usually stop there and take things from there to Europe. So Edward Gray describes some species from there. Fischer from Germany, also Peters, famous Peters from Berlin Museum, also described two important and very iconic species of frogs from Principe. Richard Griff, also German, did the first checklist of the species we know from these islands. And then it started the Portuguese exploration of these islands, and with Adolfo Moller from Coimbra, that went to the islands and got some specimens studied by Bedriaga. But the biggest expedition we had in the 19th century was from this guy, Francisco Newton. And he was hired by the Lisbon Museum to do this expedition just to know the fauna of these islands, and he stayed there for a while. And given his collections, there was a lot of species that were described. So the base of our knowledge on these islands stems on the collections of Newton. After that, we had Leonardo Fea, Italian explorer that collected things for Genoa, but that were later studied by Bollinger from the British Museum. We also described some other species from these islands, namely some blind snakes and colubrids and lamprophets. And Miguel Angel from Paris Museum also described some typhlopids from here. And then already in the 20th century, there was some other expeditions to the island, namely by the Portuguese government, which resulted in these two checklists by Sara Manassas, which kind of stayed for a long time as the only checklist we had for the country. Also, Lilia Capucassia worked on some snakes from these islands that were collected by Fea. So we had to wait until the 21st century to start revisiting this herpetofauna. And it started with José Jesus from the University of Madeira, PhD thesis. We explored a lot of these groups and provided the first molecular data for many of them. So we started to work with Afroblepharus, now Panaspis, Hemidactylus, skinks, then Mabuya, now Trachylepis, Lygodactylus, and snakes also. So these were the new vague of study of this species. Then, of course, a really important contribution was by the team from Bob Drews from the California Academy of Science. We had these Gulf of Guinea expeditions, and we explored the islands for a long time, and they're still doing it. And their main contributions, of course, they collected dozens and dozens of species of reptiles. Their main contributions were mostly on frogs, and they described some new frogs from these islands, but also some geckos that I'm going to talk further ahead. And some of really important papers about the biogeography of frogs here were produced by Ray Nabel, working with Bob. about how the frogs got to the island and speciated there. So this was recently described, Pyrolis druesi from Principe Island. And one pattern that we see in frogs, but then we'll see also in reptiles, that what we have in one island is different from what we have in the other one. And sometimes they are sister taxa, sometimes they're not, even if they are in the same genus. Then I started to work on the islands about 7 years ago. And I focus mostly on taxonomy, so I'm mostly a taxonomist, and I started to work on land animals. And the interesting thing is, besides I'm a herpetologist, my first species description from these islands, but also from ever, was a shrew from Principe. But since then, we have been working on Trochilipes. We described 3 new species of Trochilipes. We described a new species of a cobra, and we described some Panaspis skink. And as Crystal was saying, there is not many checklists about the herpetofauna of islands, but we recently made a provisional one for São Tomé, Príncipe, and Annobon, but it's already outdated, as I'll show. So an overview about these islands, about numbers. So Principe Island, the oldest one of the small ones, has 14 species, and a lot of them are endemic. So 5 species of skinks, 4 endemic, 4 species of geckos, 2 endemic, and 2 are introduced. 1 species of colubrid, 1 species of endemic lamprophid, the boidon, and 3 species of blind snakes, which 1 is confirmedly endemic and the other 2 are dubious. São Tomé, which is a bigger island, has less species though, but also is a younger island. So he has a Pelomeduside, Pelusius, a mud turtle, 1 species that is introduced, 2 skinks species that are endemic, 4 geckos, 2 endemic and 2 introduced, and snakes has a little bit bigger diversity with 1 colubrid endemic, 1 lamprophid endemic, 2 blind snakes that are both dubious. We don't know if they are the same of the ones in Principe, so that's why we have this in here, and 1 endemic species of cobra. So, and when we look to Annobon, which is the younger island and much smaller, we still have a lot of endemics for the proportion of the area. 2 species of endemic skinks, 3 species of geckos, of which 2 are endemic, 1 endemic colubrid, and 1 introduced blind snake. For such a small place, it's a really interesting diversity and really is from many groups. So let's just go through some of these groups. So the species that we have in São Tomé of mud turtle is Pelusius castaneus, and it was recently accessed by the phylogeny of Fritz et al. And it shows that the population that we have in São Tomé belongs to Pelusius castaneus, which is a species that is located in all West Africa, some Congo, Gabon, so all those areas. So makes sense that the thing got there. From these areas, it's one of the West African representatives of the fauna. For skinks, we have several examples of skinks in here. So this is the genus Panaspis, the small leaf litter skinks, or snake-hide skinks, as I want to call it. And we did this review, and we found an interesting pattern. So the populations from Principe and the population from São Tomé in here are sister taxa. Although the population in Annobon, it's not sister taxa with this, which suggests a different colonization, while in here suggests a stepping stone, but is sister to Panaspis cabinda, which is a species that comes from, northern distribution in the Congo and goes south to Angola. So this is it's not a typically West African species, but more like already almost southern Eastern African species. And it's interesting because Annobon, of course, is one of the southern islands, and we will see this pattern again repeated with some snakes. For Trochilipes, we did this work on 2016, and where we reviewed the Trochilipes species of São Tomé and Príncipe. And what we found is that the species that was considered to occur there was Trochilipes maculilabris, which is this big complex that goes from West Africa to East Africa. We already know that's a species complex in the mainland. And what we found is that the population from Príncipe, which is the closest to the coast, is a sister to the maculilabris, true maculilabris from West Africa, but it's different. While the Trochilipes maculilabris from São Tomé, which is the other island in here, it's not sister to maculilabris itself. And as you will see, it's sister to a thing from East Africa. And of course, we had Trichylepis affinis, which is also another species complex that occurs mostly from here to here, and our population fell within it. But more recently, there was a bigger study on Trichylepis by Jeff Wynell et al. That looked a little bit again more to all Trichylepis species of Africa. And what he showed is that the thing from Principe, of course, it's more related to the Maculolabris. The São Tomé species is more related to the Maculolabris clade that is closely related to the Comoros and to the East Africa. So that's an interesting pattern there. He also showed that the endemic Trichylepis from Annobon is more closely related to the West African thing. So Annobon, as we are already seeing, as species, the endemic species that are related to West Africa, but are also things that are from Southern Africa, which suggests multiple colonization events from the different groups. In 2015, I described the species from an islet in the coast of Principe, and this is a really interesting case, and also I got a big lesson in here: never trust morphology alone. So when I got this specimen from this islet in the coast of Principe, so it's like this islet in here, I only got it from an old specimen from a museum from the 70s. And it was formalin fixed, and I couldn't extract anything from it. I tried several things, but I couldn't do it. But morphologically, it was completely different from anything else, from what we had. We described this as new because it was much bigger, the scale counts were different, coloration was different. But recently, I visited the island and I put it in the phylogeny, and what we saw is that it's the same thing as the thing from Principe, even about the size is different. And with the collaboration of SITS Norder, which is here, we started to model the sea level changes in the Gulf of Guinea, and we saw that in the last million years, the Tinhosa Islet in here was connected with Principe for several times. And our mitochondrial data suggests that this is a really recent split between these things, which is basically at the same time as the last glacial maximum. The same thing happened with the Hemidactylus from Tinhosas and Principe. So in São Tomé we have this, and the Gulf of Guinea, we have these species from the Afro-Atlantic group, and again we have species that occur in both islands, the one species in each island which are our sister, and other species that were introduced, but all belong to this African Atlantic clade. So in here you can see well, but this is the African Atlantic clade of Hemidactylus, and the species from São Tomé are within it. And also there are species that were introduced that come from Angola that occur in São Tomé. I know I'm almost going out of time. So Lygodactyl is another species of another group of geckos, and what we have is that we have sister species, one in each island from Principe, São Tomé, and Annobon. Again, for colubrids, a recent paper by Engelbrecht et al. That looked to Philotemnus show that the things from São Tomé and are within this group more from southwestern Africa, and the things from Annobon are really close to things also from Angola, which suggests again a colonization from this side instead of from West Africa. For Apsidophis or now Gastropixis, we don't have much data. We know that the thing in Principe is endemic, closely related to Smaragdina, which is a West African species. For Boydon, we have one species in São Tomé, and it was thought to be the same in Principe, but ongoing work showed it's sister taxa, so it's a new species from Principe. For blind snakes, we know that we have two valid species in São Tomé, these two ones, and these two ones were described from Principe, but we don't have molecular data yet to see if they are the same thing or not. But these are mostly related to West African species. We also have this guy in here, this Typhlops elegans from Principe, mostly related with West Africa. And then the thing we described recently, the cobra. So there was a cobra there in São Tomé that everyone said it was an invasive species that was introduced by the Portuguese. But this never made much sense to me, because first of all, why would they introduce such a snake, such a cobra, in the place you live? To control rats, they said. But the thing was that it also makes sense was the fact that the first Portuguese that arrived there already talked about this cobra. So when we did a phylogeny of it, we saw that it was different, with sister to the thing from West Africa, but was new. So we described it as new, and this was an interesting thing because sometimes without poor taxonomy, we can start claiming that it's an invasive species, but in fact it's endemic, and you could have wiped it out trying to get rid of it. This was then followed by a new paper from Wolfgang Museu that confirmed that our species is sister to the West African cobra. So I think I already took my time just to say that there's ongoing work on São Tomé, and we really need to fine-tune some of these phylogenies to make a general pattern of colonization of these islands. But what I wanted to say is that island reptiles need more study and also conservation efforts. And with this, thank you for assisting. Thank you.
There's time for a question, not a long one. Short one. Actually, I have a comment, if you don't mind. That it's a good example of reptile diversity just skyrocketing on an island system. Just because thank you. Just because you actually have done surveys there. So all of these non-surveyed islands or not well surveyed could have it could be the same effect. So quite cool. Thank you. Oh, you do have a question.
So it's actually a question to the both of you. So you both talked about these ocean currents and about the rafting, and I was wondering, so these are the currents, right? So what about the past? Weren't the currents in the past different from what they are now?
Well, I think, so I didn't add much research on it, but for example, Benguela current is a really old current already. So, in fact, it's a thing that we should take a look.
Yes, and they were. And if you, our next speaker, Jason Ali, has done a study on exactly that. So looking at paleo currents, and you can then match that to dated phylogenies to see if the movements are in the opposite direction of what you would expect. So I don't know about Gulf of Guinea though.
Yeah, I have to take a look.
Yeah. Thanks. Okay, so now we can have our lone geologist, Jason Ali from University of Hong Kong, and during question time, please, he's never had biology. You didn't have high school biology, you said. Yeah. So you can ask him questions about the volcano, but but maybe not about biology.
Okay, what I'm going to do is present some work that has just been published in Ecological, and the critical thing here is this element of the title on volcanic ocean islands. So we're actually going to look at something, and we've got a very specific island type that we're looking at. Okay. Now, in 2014, I went to the island biology meeting, and the thing that struck me was the enthusiasm that people had for this thing called the general dynamic model. And it seemed to me that everybody was going crazy about its potential for explaining lots of things biological. And whenever I see lots of people who are sort of all shouting from the same hymn book rather vigorously, I tend to have a certain degree of skepticism. And because I'm a geologist, I also understand that the issue of the island's construction and the way it's actually shaping the biology, there could be some problems because islands are in fact rather complex from a geological perspective. Right, okay. So in order to do this, I thought about which sort of organisms we could use to test these, and there are various sorts that we can look through, but it so turns out that the reptiles are the optimum ones for various sorts of reasons, and I don't really have the time to go through those, but really want to stick with reptiles because of one or two key features. Now, I started to do the work, and the problem with being a geologist is that sometimes you run into a brick wall because I don't have that biological training. So I tried submitting some work to a couple of journals and got smashed. And at that stage, as I do normally, because this happens quite regularly, I sort of sat there and sulked, and eventually I thought I must try and resurrect this. And then in 2017, Shai Miri actually wrote in Frontiers of Biogeography a rather damning view of studying the biology of volcanic ocean islands. And he says, I claim that oceanic islands, especially the archipelagos of Hawaii, Canaries, Azores, and Galapagos are essentially highly overrated. And when I saw this, I got onto ResearchGate and I said, that is the paper that I would have liked to have written, you cheeky person. Anyway, we sort of left it at that, and then a few months later I decided to contact Shai and said, look, I'm working on this particular project to try and look at this general dynamic model, particularly this issue of in situ speciation. Would you like to join me? And he said yes. It comes over to Hong Kong, and we set off, and the relationship's just started. And then he reveals, shortly after he's arrived, that he's got one of the world's biggest databases for reptiles. Basically, he's got any reptile that exists, he knows where it lives. And so it's a bit like going and starting a relationship with somebody, and then suddenly finding that their family are like billionaires. So rather good. Okay, so this is the data set that we're using, and we're focusing very specifically on volcanic ocean islands. I don't want to confuse any other island types. And we've got islands, 53 islands from 8 archipelagos, and there is one single island which is, is up at Cocos, which is just off Central America over here. And altogether we've got 194 species, and there are 270 species island records, and then we have 144 species are single island endemics. And we've got all the way from the Mascarenes all the way across into the Pacific. And when we look at the data sets, then we've actually got, the actual individual islands over here, and then we've got some geographical information, and then we have our species lists, and then we try to work out the relationships between those particular species. So we can see that we've, in our blue colors that we're going to use, then we have, these are non-sister congeners. The orange are to denote species radiations. We've only got a couple of those. And then we have the green records, which are records of in situ cladogenesis. And then we have the yellow records, which are questionable records. We don't really know because of various reasons. And then we also have, I think on the next slide, we have 1 or 2 species which are found outside the archipelagos. Okay? The other key elements in here are A, B, C, D, all the way through to O over there, represent the different clades. So these are effectively the primary colonization events. Okay, and we can see the key message here is that in situ speciation is very, very low. We have 2 radiations, the tortoises on the Galapagos and the Phelsuma geckos on Mauritius. Then we have a small number of cladogenic events, and some of them are questionable as well. Okay, so we then have to test them against some sort of various physical, so we've got very low levels, but we need to see if there's any sort of relationship by physical control, so we can look for these in terms of area, age, elevation, and remoteness or isolation of the island system. And we'll show you a few plots on the y-axis. On the x-axis, we're going to plot the physical property. On the y-axis, what we're going to do is for each island, we'll show you the proportion of the total reptiles on the island which result from in situ cladogenesis. And then, because we have both probable or certain and possible, I have 2 plots for each property. Okay. So we can see in this particular instance, first of all, we can see the large number of dots which are sitting at 0. So in this case, probable cladogenesis occurs on 6 islands. And we can see in this particular instance, around about 40% of the species today result from that process. The real issue here is that it's very, very low levels. And we can then go through, if we then add in the possibles, we can see our data set increases slightly, and we get also one of these points coming up to 100%. I think this is on Rodriguez Island, just up across the sea slightly. And then we can do the same things for age. Now, the key thing about the age is that there's no, in the general dynamic model, the key prediction is that there will be a spike in situ cladogenesis or speciation taking place somewhere probably around, we'd expect to see the, right, okay, is that age? Yeah. Oops, it's died on me. But we would expect to see some sort of hump shape pattern in here in terms of the age, and we're not actually picking this up, so we can sort of, it's actually taking place in some very, very young islands, and it's also taking place in some very, very old island systems. Okay, and we can look at elevation as a proxy for, use elevation as a proxy for habitat, And again, but again, the key issue is because we have low levels of in situ cladogenesis, it's not really going to show through. And then we can go through, Shai then does the statistics on these. I'm not trained in this area, but he then runs a whole series of statistical analyses using a couple of multivariate models. And I can't possibly explain these to you because it's not my background, but you can read the paper or you can send an email to Shai. The other thing that we did was we wanted to check, or I suggest that we check, was if the presence of a non-sister congener on an island was also inhibiting speciation. It could be that basically you've got something quite similar and it sort of stops you from doing it. But it turns out if you run the tests on this, statistical tests, then this is not valid. Where you've got more chance of being able to have cladogenesis when you have a non-sister species present. Okay, so in terms of biodiversity, then we can rule out there's three elements which control biodiversity, and we can effectively say that biodiversity is increasing simply due to immigration, and it falls due to extinction, and we can rule out the issue of in situ cladogenesis effectively. Okay, and what are the reasons for this? Well, our volcanic ocean island systems are very complex. We can't reduce them to a simple plot, which is what the Whittaker model suggests. There could be biological elements which are causing this. The other chief issue is the fact that the islands are not big enough. There simply isn't enough space. And the other issue that we've got to think about is the idea that our islands are actually forming in different sorts of tectonic settings. So we've got the hotspot islands moving on the fast plates. And these are actually the ones which are going to have the island development, the ontogeny model, which is closest to what the Whittaker model says. On the slow-moving plates, which is what we're on at the moment in Africa, we're traveling at around about 2 centimeters per year. And we can see that we've not only got two volcanic massifs here, okay, so you've got a very, very young system next to a somewhat older system, so our physical model's not really going to work. We've also got some island groups in highly unusual tectonic settings. The Comoros Islands are very, very strange, as are the Gulf of Guinea Islands. They are not on a hotspot. They are developing in different ways. And then some of our island arc islands, which are over in the Lesser Antilles, which is what Graham will be talking about, they're in areas where the volcanism is quite different and it's highly explosive. It's nothing like the rather effusive volcanism that we get over here, above a mantle plume hotspot. There we've actually got a completely different volcanic system. Yes, it's a volcano, but it's behaving very, very differently to the volcanoes that we have in this part of the world. Okay, so at this stage, we're sort of getting through to the paper and we're sort of saying, well, how do we actually move forward on this? We don't really want to end on a negative, or Shai just says, oh, we knew it wasn't going to, we sort of knew it wasn't going to work and it hasn't worked. Yeah. Great. And I said to him, no, we've got to try and explore the immigration element to this. How does the immigration into the island work? And the thing that I noticed was that if these are species lists, I think the top one is for the Cape Verde group, and the critical thing to here is to recognize the fact that we have just 3 clades, A, B, and C. Okay. And if we go through there, we've got 21 species in 3 clades. But if we come across to look at the Gulf of Guinea Islands, okay, then what we have are 23 species in 14 clades. So the migration and then what happens subsequently is quite a different beast. We've actually got to start looking at these islands in a different way. Okay, and so what I've got on here is for each of the islands, this is the Cape Verde group, we've got 3 clades, and I've just ranked them 1, 2, 3, and these are the number of species in them. So our most species-rich clade, which is going to be plotted at this side of the plot, here has got 12 species, then we've got another one with about 6, and then another one with about 2 or 3 in there, and we can come across to the Canaries showing something similar. And then Galapagos, again, they're all around they're typically between about 9 and, the richest clades between about 9 and 15, 14 species. If we come to the clade-rich island groups, then they have very, very low levels of speciation have taken place. The maximum is 4. You can see many, many clades, but they're all relatively poor. Okay, so what are we doing for time? Right, okay, we're all right. Yeah, okay, so this process, in terms of colonization, the process is going to be enhanced when the journeys are short, when the passage is relatively easy for the animals to go through, and we also need a supply of rafts to get the things over, and we also need conducive surface water streams, and we also need our target land masses need to have appropriately climates and habitats so that the animal can then establish a new population. Okay, what we find with and I don't want to go through some of the stuff in the lower part of the plot, but we have relatively few clades. As I've been through this earlier on, okay, and in these instances, we have clay pore assemblages. They're difficult to get to. They're very large distances. The only anomaly is the Canaries, but separating the Canaries from mainland Africa is the Lanzarote. There's a current running through the Lanzarote Gap. And this is, in fact, ring fencing, or very efficiently, the Canary Islands from West Africa or the Iberian Peninsula. And the other thing as well is that with Galapagos Cape Verde Canaries, we've also got fairly hostile environments, very, very dry, so it's difficult for them to get off. Okay, if we come through to the clade-rich assemblages, and we don't need to go through that, but effectively the island systems are less isolated, typically 200 to 300 Ks. They tend to have relatively wet climates. They are prone to storms, so we can get these rafts off these land surfaces, and also the journey should be easy because we can basically hydrate the animals as they're coming over. Okay, right, so what I've done here is I've turned these diagrams that we had, and I've turned them into effectively like silhouettes. So come back, and we can see over here that we've got, these are for the clade poor groups, these are the clade rich, and I've just used this rather than drawing the specific plots. And we can see these two different types, and we can just refer back to these diagrams over here. And then what we can then do is to at least throw into the air, I'm not sure how some of these predictions will work, but we can then start to look at the responses or potential responses for the mammals, the land mammals, as well as the amphibians. And we can then possibly think about how the flying organisms can get there as well, the bats and the birds. And we can see these dark patches indicate non-endemic species, so we would expect to see a fairly high proportions of non-endemics coming in with the flyers, and zero or very, very low levels with the organisms, the land organisms, which find it difficult to track over. Okay, right. So in terms of what we'd like to do, or I think areas which you can perhaps follow up on, are extending this work to look at other island systems with these looking at the other volcanic island clusters. I suspect that the clay poor systems are relatively rare. I think the bulk of them will be clade rich and species poor. And we can also then extend them to the non-volcanic ocean island archipelagos, and then we might then want to start thinking about how other organisms fit into this. So that's it. You've got some nice lava lizards. Thank you. That's 15, right?
Thanks, that's really interesting. Richard Field from the University of Nottingham. You talked about early on about the reptiles being a close to ideal group, but it's kind of skirted over the reasons why. And then it struck me that they're a pretty good group for ruling out in situ cladogenesis. Yeah. But does that make them ideal? So perhaps you can just think about what the advantages of working on reptiles and versus other groups.
Right. If we're looking specifically at single islands, which is what the GDM say, we don't really want organisms bouncing around within an archipelago. Okay, so we want the stuff can just about get to islands, but we don't want them to be zipping around willy-nilly like bats and birds as such. So, and the other reason is we've got good phylogenetic data, so we could perhaps try this with some of the invertebrates, but the reality is that we don't have the same level of coverage, and obviously the idea for doing a presentation like this is to try and encourage people to look at which organisms might fit the model. As is, we just assume it works, and it's not been apparent from me how rigorously anybody's tested it. And I think we've made at least some effort to try and sort of say, well, let's look at one group of organisms and see if it works. We don't think it does. It probably doesn't work for the other terrestrial vertebrates. So what organisms are going to fit into this? Yeah. Okay.
Yeah, thank you for your very nice talk. You ask what type of organisms fit the GDM, and of course your work is on islands, so you can also ask what type of islands would fit the GDM. And I was curious because in your plot you showed all the islands for which you have the data in a single plot on H on the x-axis and on the y-axis. What was it? Can you go to the slide?
I'm not sure if he's locked it. Sorry about this. Is it these?
Yeah, that one. Yeah, so I would. So yeah,
elevation, yeah. Do you want age?
Yeah, exactly. So well, I would be curious. So here it's indeed that the number of clades with let me think little. Well, I was just thinking if you would make this plot only the data which have a large proportion is really small, but I would be curious because for Hawaii, if you in this one archipelago, I would expect that this relationship was more evident than for other archipelagos that do not follow this progression rule. And as you so what do you think about that?
Yeah, but we're not done Hawaii because Hawaii is not.
Yeah.
Yeah. I don't know. I mean, we've got to go out and find more data. I don't think there are many islands. I think we could fool ourselves by telling us there are lots and lots of islands. There aren't that many. When you actually start to look at it, there are not that many, and they become very, very complicated geologically. As I say, I think most people would assume that Gulf of Guinea is a very, very simple way to form them, and they would call them a mantle. But it's not. If you look at the geological literature, they're completely anomalous. So you've got. I think this is the thing. I think we need to get more geo.
I believe you. But then now in this plot, it's all in the same plot. But if you would look at a single archipelago, then we could say, okay, it works for this archipelago, but not for this archipelago. I mean the GDM. And I fully agree with you that we need more geo. So I'm just curious if we would do that. So for which archipelagos it would work?
But the numbers are so low that there's nothing. There's nothing that really struck us. It wasn't obvious. Yeah.
Yes, that's yeah.
Okay. Right, thank you. There's one more. Okay, sorry. Oh, it's Anthony. Oh no.
So here you're looking just at these oceanic islands.
Yeah.
And so you hit upon one thing is that area.
Yeah.
Most of these are very small.
Yeah.
And I think, isn't that just one thing that is limiting in situ cladogenesis?
Yeah, of course it is. Yeah. We just don't, but that's the reality. We've got this model, and people love it because you create islands from scratch. But in reality, at least for these organisms, they're simply not big enough. And we're doing some work with another group of people who've worked on mammals. So we're doing looking at all sorts of different islands. But you probably need to get to about 10,000 square kilometres before it really starts to kick in.
Okay, great. Thank you, Jason. Okay, so our next speaker is Graham Reynolds from the University of North Carolina.
Hey, good afternoon, everyone. Can you hear me? Is this working? Yeah, good. All right. Well, first, I just want to say thank you to Crystal. It's really a great pleasure to be here at my first island biology meeting, so I'm really excited. It's been a great week. Really, I've loved all the talks, so thank you for the invitation, and thank you for to the conference organizers. What I want to do today is tell you a little bit about some of the work that I've been doing recently. And I want to do two things, really. I want to convince you, one, or maybe remind you, that the Caribbean is an amazing place, and we still have a lot to learn about this region. Okay, as one of the few Caribbean representatives here at the meeting, I want to emphasize that. And number two is that a lot of the stuff that I'm going to show you, the kinds of methods, for example, can be applied across a bunch of different taxa, not just reptiles, but also pretty much anything. So I hope you'll be able to take those away from this. So I'm going to talk about ecological and evolutionary determinism. And I want to start off by saying ecological and evolutionary. I'm not necessarily wanting to partition those two things apart. Okay, I'm really thinking about the interplay between ecology and evolution and how we can understand how those things work together to produce the outcomes that we see. So it's about pattern and what are the processes that underlie those patterns. Okay, so here's what I'm interested in. The production of the phenotype, how that phenotype interacts with the abiotic and biotic conditions in which it finds itself, and also how those interactions influence evolutionary trajectories on islands. And so in order to do this, we need to understand the interplay between ecological and evolutionary processes to characterize what we might refer to as determinism in the evolutionary process. In other words, predictability. And I'm using that in a way that's contrasted with stochasticity or randomness. Okay, and so we don't always expect determinism. We don't necessarily expect it. We certainly don't always see it. But when we do see it, that's fairly didactic and fairly exciting. Of course, a classic example of this would be the Caribbean anolis lizards in the Greater Antilles. Now, let me just pause for a minute and say, if we think about the patterns that we're talking about, these patterns that we observe, what are the processes, the evolutionary processes that underlie this? Well, maybe you can say them with me, but we know the four big processes: mutation, selection, drift, gene flow. Okay, we can think about which of these might expect to produce stochastic versus deterministic outcomes. Okay, so mutation we might think of as being somewhat deterministic. We have mutation rate matrices, we have third codon mutation rates that are different from first and second, so forth. There's some determinism there. Genetic drift, stochastic, right? At least we think it is. It's sampling error. Gene flow, fairly stochastic. Of course, there's things that are going to be driving gene flow into island systems. But then there's selection. Okay, and selection can produce these deterministic outcomes, and that's what I want to think about today. So here's the Greater Antilles to orient ourselves. The Greater Antilles in the northern Caribbean. This is a map showing some of the field sites where I've been working the last few years. But if I were to map onto this, the field sites where all my collaborators have worked, you would see just would be covered. So a lot of field work in these amazing places in order to get the data that are going into the analyses that you'll see momentarily. So I want to start off by talking about this classic example with the anolis ecomorphs. And what we're talking about here is that on these Greater Antillean islands, we have radiations of anolis lizards, and these anolis lizards occupy what we call these ecomorph phenotypes. In other words, they are specifically adapting to certain aspects of the environments on these islands. And so we have things that we call trunk ground anoles, trunk crown anoles, trunk anoles, twig anoles. Okay, these are ecomorph categories. And so we have this classic idea about the evolution of these ecomorphs. This is coming from Williams in 1969. These ecomorphs occupy different levels of, say, a forest canopy. Okay, and they have different associated phenotypes with occupation of these different habitats. And what's amazing about this system, of course, is that we see repeated evolution of the anolis lizard ecomorphs across the Greater Antilles. So we see an assembly of anolis ecomorphs on Cuba, on Jamaica, on Hispaniola, and on Puerto Rico. The same kinds of ecomorphs, right? This is determinism in the evolutionary process. This is a pattern, an outcome of selection resulting in these phenotypes that are repeated. Okay, so that's what I'm referring to when I say determinism in this context. So we might ask ourselves, okay, we got these deterministic outcomes in Caribbean reptiles. Are there other groups that might demonstrate this? And so this notion that Caribbean reptiles might have these sort of repeated patterns of evolution or determinism, and particularly in the context of this sort of catchphrase, adaptive radiations, is an intoxicant. And I say that because, this is something that a lot of people really want to test. Like, well, why don't we just see? We got this radiation. We got some different morphology, some different phenotypes. Let's see if it's an adaptive radiation or if it's some way deterministic. And so we might say, well, a few of these groups have been looked at, and we find a mixture of sort of maybes and no's. So the maybes might be the spherodactylus geckos. The ones I say nope to are the dipsodine snakes and the cyclura iguanas. And so cyclura iguanas, there's a beautiful picture there. Here's some pictures of the spherodactylus geckos. Okay, I just want to show you some beautiful creatures here. So maybe, probably not on some of these. In other words, people are testing this, they're not coming up with much, but that doesn't mean necessarily something important, right? We don't necessarily expect this to be the case for all different groups. That's okay. Well, I'm going to focus specifically on thinking about the Greater Antillean anoles and the Greater Antillean boas. Okay, it's almost like a predator-prey relationship. But in particular, I'm going to think about some aspects that have not been looked at in these two groups. So let's start with the boas. This is an amazing radiation of boas in the Greater Antilles and Northern Caribbean. 13 species, all different kinds of phenotypes. They live in different habitats. Some are arboreal, some are terrestrial, some are, live in both areas. Some are huge, some are tiny. 13 species, and yet we see this incredible radiation. Here's where they're distributed. A number of the areas in the Greater Antilles and then the Northern Bahamas. And when I say things like big and small boas, I really mean it. Take a look at this. This is the largest species of boa in the West Indies. It's the Cuban boa, 4 meters long. That's an adult female that my friend Pete Tolson's holding. Then we have small species. Take a look at this one. This is the Hispaniolan vine boa. That's an adult female. Okay, so we're talking about 13 species, a rather modest radiation, and yet we see pretty significant phenotypic differences. So we talk about this in the context of exceptional morphological and ecological outcomes. This is another cool example. This is that Hispaniolan vine boa that looks a heck of a lot like a mainland colubrid snake, which is this blunt-headed vine snake. They look the same, they do the same kind of thing, they feed on the same kinds of things. It's incredible. Okay, so exceptional morphological and ecological outcomes in a modest radiation, 13 species. So one of the questions we wanted to ask was, do we see determinism in the evolution of body size? In other words, when we go to islands in the Greater Antilles, we see big boas and small boas, and in some islands there's both. So we might ask ourselves, well, is that a product of a deterministic process, or is it just random? Let me simplify that question a little bit more. Did small body size evolve once and spread to the islands, or did small body size evolve over and over? That's the question we're asking. Why might we want to ask this question? Why might we expect it? Is because we've seen this kind of pattern in other groups. Okay, we might call this the island rule in insular reptiles. Okay, we see this in a few groups, not all of them certainly, but some. This is a result of determinism and convergence. And a great example of this was a recent paper published about Boa imperator, which is a Central American boa, on islands off the coast of Central America, where you have small islands, and these small islands have been colonized independently by mainland boas, and on each of these islands, the boas evolved small body size. And this is probably an adaptation to two things. One, arboreality, and two, feeding on a specific prey base, which is migratory birds. Now, it's important to keep those two things in mind, habitat and diet. So what do? Is we build a phylogeny, all these species, we look at the different body sizes, and we ask ourselves, when we look at this phylogeny, do we see the evidence that small body size evolved once or multiple times? And what we find is that there's a minimum of 4 transitions in body size in the evolution of this group. And so we're starting to go, maybe this is suggesting something regarding deterministic outcomes. Okay. And so we can see this perhaps a little more clearly if we paint those body sizes onto the phylogeny of these boas. And now we can see very clearly that this phylogeny, this quantitative trait, just simply body size, does not show any phylogenetic signal. In other words, it hasn't evolved just once. We see it evolve over and over again. Check it out. Small body size evolved up here. These are 2 species from Hispaniola. Small body size down here, Puerto Rico. We've got giant boas right here. That's the Cuban boa. Okay, it's kind of a mixture. Okay, so this is leading us to understand that small body size has evolved over and over again on these different islands. That's determinism. Now we take this a step further. We want to ask, okay, well, if we see the evolution of small body size, is it related to any sort of ecological characteristics? And so we have this other incredible situation, which is that some of these boas are specialists. And they're specialists in 2 ways. One, habitat. In other words, some are strictly arboreal, some are terrestrial, some are generalist. And diet. Some of them specialize on diet, in particular on anolis lizards. You guessed it, right? Okay, so we can ask ourselves, is there correlated evolution between body size and specialization? And this is a bit of a tricky thing to do. What we're doing here is trying to model the simultaneous evolution of discrete character states, in other words, are you a specialist or generalist, and a continuous character trait, which is body size. And so this has been challenging to do until fairly recently. What we did is we're borrowing a model from quantitative genetics. It's called the threshold model. And I want to show you, I just briefly am going to mention this because it's really applicable to a lot of different data sets that have both discrete characters and quantitative characters, or continuous traits. Okay? So the threshold model is basically asking, if we expect that a discrete phenotypic trait, say being a specialist, is underlain by the evolution of lots of quantitative traits, that we can model that. In other words, we can reconstruct the ancestral history of those discrete character states by modeling the underlying quantitative traits, which we call liabilities. So that's what we're looking at here. What you're seeing in this example of how the threshold model works is you're seeing a Brownian motion model, and you're seeing a flip between character states, and that's the changing of the colors. Okay, so this is what we're modeling through time, are these underlying liabilities, are they causing these different lineages to flip into a different character state once they hit a threshold? So we're using that model to ask this question: do we see a relationship between body size and specialization of both habitat and diet? And the answer is, sort of. Definitely for habitat specialization. Okay, this is showing correlation between those liabilities that we model under this threshold model using a Markov approach. And we can see that small body size is associated with habitat specialization. In other words, if you get small on these islands, you're specializing in habitat. You're becoming arboreal or terrestrial. That tells us something interesting about this process, doesn't it? I mean, that's the pattern that we see. We go out in the woods and say Hispaniola. We say, oh, look, there's a small boa, and it's in the vines. There's a big boa, it's everywhere. Well, there's a reason for this. What we're trying to do is understand the process that generates this. Now, with dietary specialization, not such a strong signal. Now, there's two reasons for this. One, dietary specialization in boas is hard to quantify for some species because some of these boas are really hard to find. And if you do find them, you don't want to just cut them all open until you get a sufficient sample of diets, right? That's not very ethical. So we don't have great data for the diet, but there is a suggestion. It does seem like small body size is also associated with dietary specialization. Okay. Now, things have changed a little bit recently because 10 years ago, we only had 9 species of recognized boas in this genus. We've seen a 31% increase in the recognized diversity at the species level. Okay, so we're having to play catch up with these data analyses, in terms of the new species that we're describing. I'll show you 2 recent ones. This is the Conception Island silver boa. Okay, we found this animal in 2015. We described it in 2016. Let me pause. I don't know if I have enough time to do this. But any of you working on snakes, lest you're concerned about whether or not people actually care about snakes, rest assured they do. The news of this discovery went around the world, hundreds of articles, 6 languages. People got excited about it, and I think that's interesting. And it turns out that's crucial because there are 135 plus or minus 30 silver boas left in the world. That's it. This is the most endangered boa globally, by far. So this is a big situation, something that we're working on, doing a lot of work on right now, trying to figure out how to save them. Okay, Conception Island silver boa. Oh, by the way, that's an arboreal specialist. And then a really new one, the crooked Ackland's boa. This one we found just a few years ago, 2017, and we've only seen 3 juvies and an adult. So we don't know anything about what they do. But at least the silver boa does support these ideas about specialization. Now, let me flip to the anoles. Okay, we've already talked about how the anoles are a great example of this adaptive radiation idea, this sort of understanding the processes that generate the pattern. I want to focus on the Cuban brown anole because one of the questions that hasn't really been addressed is what's happening at the initial stage of an adaptive radiation? In other words, what did the ancestors do? Well, presumably the ancestors are moving around to these different islands. What happens when they get there? Well, if you hit fast forward enough, you think, okay, well, ecological speciation generates these different outcomes. Okay, that's sort of the idea. But what we want to look at is right at the beginning there. And happily, the Cuban brown anole is a great thing to test this on because widespread anoles are not common. Most anoles are endemic to an island or an island bank in the Caribbean. There's hundreds of species. Okay? But there's a few species that are widely distributed, and the Cuban brown anole is the most widely distributed. Okay? Ancestral in Cuba, but has then spread to all these different islands in the West Indies. So we have this incredible natural experiment that's happened. We have the Cuban homeland, and then this diaspora that's moved away from Cuba to all these different islands. So we sample around all these areas. Look at what happens to the diaspora from Cuba. When you leave Cuba, if you're a brown anole, that is, you get bigger. You increase in body size. Okay, so there's something going on here with as these animals are departing from the island of Cuba, colonizing other islands, something is happening in terms of their phenotypic evolution. In this case, they're getting bigger. They're doing other things too, but that's one example. If we look at a suite of quantitative traits, 10 in this case, and I don't know if Marta is here, but Marta was responsible for generating a lot of these data. She's a co-author on this project. What we see is that these anoles, they evolve directionally. In other words, I can take an X-ray of an anole, of a brown anole, from a diaspora population and from a Cuban population, and I can tell you, using this method, which population that came from without knowing that a priori, just based on these 10 quantitative traits. Okay? So in order to demonstrate that, we show that using, for instance, this is an example, this is a linear discriminant function analysis. I can use a machine learning algorithm to demonstrate that 93% of the time I can correctly assign an anole to either a diaspora group or the Cuba group. So again, they leave Cuba and they're changing. Now, you might notice this little hump. That's Jamaica. And Jamaica was only recently colonized by Anolis sagrai. That's basically Cuban Anolis sagrai. Okay, so that just sort of demonstrates that. But Cuban diaspora have different evolutionary origins. In other words, it's not like a few of them left Cuba and then hopped their way around the Caribbean. Instead, they have a tremendously diverse evolutionary background. The largest or deepest divergence in Anolis sagrai is between East and West Cuba, 5 to 7 million years. Then we see from both East and West Cuba populations of Anolis sagrai leaving and colonizing different parts of the Caribbean. Are you starting to see the story here? Is that when you see this sort of concerted phenotypic evolution against differing different backgrounds, different evolutionary backgrounds, genetic backgrounds, that might suggest that something interesting is happening here. Okay? So Cuban diaspora evolved directionally, independently. That's homoplasy. So there's something about being an anole, colonizing new habitats, that you evolve this colonizing phenotype. And what's different about you? You're bigger, and you have more lamellae, which are the toe pads that are used for clinging and are associated with a lot of the ecology of this genus. So we're seeing not only a shift in the mean of these traits in the diaspora populations, but also an increase in variance. And guess what ecological speciation predicts? An increase in variance under an ecological release scenario. Okay, so it's a shift in the mean, also broadening the variance, and this is interesting that this is happening convergently. So, beginnings of deterministic outcomes. Perhaps this is determinism in the early evolution of these adaptive radiations. Now, I'm not going to say that this sagrai is going to produce an adaptive radiation, but the idea is there. What's happening at the beginning of adaptive radiations? Who was the original colonist? Okay, and it suggests that ecological release and ecological speciation are contributing to this. Of course, we know that, but we want to know what's happening at the beginning. And of course, we can predict that this might then follow. These two scenarios might follow. Okay, with that, I'll wrap it up. I always finish my talk by acknowledging my lab. I have the most wonderful students. I know you all have great students. I have great students doing all kinds of great stuff. And of course, I have a lot of people to thank. These are my co-authors on some of this work who have done a tremendous amount of this work, and as well as field assistants. And of course, these kinds of projects where you get to traipse about the Caribbean are expensive. And so we have to acknowledge the funding, generous funding for all of these different things. And with that, I'll say thank you. And I don't know if I have questions.
We can also do more questions at the end.
Hi, over here. Thanks for a great talk. You discuss specialism and generalism at the level of species. I'm wondering if you know anything about intraspecies variation in that, and say for a generalist population could be made up of different individual specialists, or every individual is a generalist. And if you do have that information, is that related to body size variation within this generalist species?
Yeah, that's a great question. So the idea is, are we seeing intraspecific variation in terms of specialization and body size? The short answer with the boas is no. It's pretty, it's pretty clear that they're doing the same thing, and that, on average, their body sizes are about the same. Now, I'll say one thing is that there's one species of boa that has tremendous sexual size dimorphism. Males get to a maximum of about 70 centimeters. Females get to almost over 1.5 meters So we're talking about, and males get to like 80 grams max, females get to like 2.5 kilos. So ecologically, those two are quite different probably. We haven't looked into that yet, but that is an example of sexual intraspecific variation. So that is one example, but yeah, that's a great question.
Okay, cool. I think we will save questions for the very end if you have more, and call the next speaker, Anthony HERREL, who's from the CNRS and the Natural History Museum in Paris.
All right, thank you very much. All right, can you hear me? Yeah. So thanks, Crystal. Thanks, everybody, for being here. It's great to be here. It's great. I've heard lots of good talks. And so what I want to talk to you about is a little bit of work we've been doing. Even though it's my name here, I've been doing this with a bunch of people, and specifically Colin Donoghue, who's been a leader on this project for the past 6 years. So if you think about island lizards, and here I'm taking you to Mediterranean islands, so a bit different. They're not volcanic oceanic islands. They can be big, they can be small, and I'm specifically talking here about very small islands. And if you go through the literature and you look at how lizards change from the mainland to small islands, there's quite a few papers out there that show that lizards on islands are different. We've talked about the island syndrome, and these things behave differently. They do different things. Just to give you a few examples, this is what things would look like on the mainland, close relatives, and this is what these things would do on an island. This is Psammodromus, closely related to Galotia on the Canary Islands. These are big, big lizards. Different colors, different shapes, different phenotypes. This is the same species. This is the same species on a mainland population, and this is the same thing on an island. So again, changes in morphology, changes in phenotype, changes in color that we see over and over again. People have looked this also looking at what happens when you go from a large to a small island, and so this is a paper that Colin did looking at Podarcis hardy in Greece. He measured lizards on all these different islands. He also looked at intraspecific interactions. And so what he showed really interestingly is that as your island here is big, as your island gets smaller, your body size increases. You also get more and more interactions between males and females on these islands. And so the general pattern is for these lizards, for these Mediterranean lizards at least, is that on small islands these things get much bigger, much more robust, they change color. Their phenotype is really changing. So in general, they seem to be darker, larger, more robust, bigger heads, larger bite forces. A bunch of them start eating different things. They become herbivorous, or partially at least. And they're often at higher density, and they're more aggressive. And so we see this kind of island syndrome for these Mediterranean lizards on small islands. And so we can ask ourselves a few questions. How to build an island lizard? How do I create this thing? Is this happening over and over again? Do we see determinism? Do we see convergence on these islands? Or is this just a random migration from one island to the next, and it's the same thing over and over again? What are the temporal dynamics? So we've talked about, Luis talked about this lizard on this island, just offshore of the bigger island, being morphologically very different. He said like blast glaciation, so it's pretty recent. But how fast do these things develop? How fast do you evolve an island phenotype? This is a really interesting question, and we don't really know. And then finally, what are the drivers of this kind of island phenotype? Is it competition, as suggested by the paper that Colin did, males fighting? If you fight, you have to be bigger, you get an advantage. If you're bigger, there could be some metabolic advantages. If you're darker, you can heat up faster. So there's potentially a whole bunch of things that could be driving these phenotypic evolution. And so how do you build an island lizard? Well, ideally, this is what Colin came up with, and I followed him all the way through. What we want to do is to do an experiment. Let's throw some lizards on islands, very small islands, and see what happens. It sounds easier than it is, but it's a lot of fun. So where do you do this? Well, we are lucky to have a very good friend and colleague, Panagiotis Pafoulis, in Greece. And he was able to help us to get permits to introduce lizards on very small islets. So most of the islets are about the size of this stage. I'll show you a couple of pictures later. And one of the conditions that we could do this is that we would be able to take them off afterwards, and that there were no lizards on the islands to begin with. Yeah. And so we took islands from our source population, a large island, the largest island in the Cyclades, Naxos, and we put them on these little small islands. So where do we go? Well, we go to the island of Paros. This is an old map of Paros. And it's really a perfect place to do this because you have all these little islets in this bay outside of Nassau. And so on these little specks of land, there are no lizards, and they fulfill all the conditions of what we wanted to do with our experiment. The other reason why it's nice to go there is because there's great food and great scenery. But that being a side element. So what did we do? So we have on the island of Paros, this is Antiparos right there. So we have 5 islands where we introduced lizards. So we're situated right here in the Cyclades. And then we also have 5 control islands. The control islands are going to be interesting because over time we want to look at what is the impact also of a lizard on, for example, arthropods, vegetation, and how do you change the community dynamics in these very small islands. This is what it looks like. It's quite nice. They're very tiny little islands. We have 5 of those, and there's a little bit of vegetation on the islands, and that's about it. So you can imagine these are harsh conditions, and the selective pressures on these islands are probably going to be quite large. This is their species. This is the lizard that we get on Naxos, on the very large island. It's the largest island of the Cyclades, and so we're going to put this guy on this island. It's kind of fun to work on these islands. How do we get to these islands? So this is Colin. I just had to show a picture of Colin with all the gear and all the stuff that we take with us to get to these islands. And we have these foldable Oro kayaks. So we take these kayaks and we can kayak every morning to our island, do our work, kayak back to the mainland. And so we're pretty much independent of other ways of transport. It keeps us fit as well, as you can nicely see. So this is fun. We can do this. We get our kayaks, we can paddle off to the islands. And so this is some of the data that we've collected so far. We started the introduction in 2014. On every single island, we put 20 lizards, 8 males and 12 females, because we really wanted to get the population going. And so these are the tallies. 2019 isn't in there. We've been back in 2019. So initially, this is 60 animals on the islands. And then every single year, we caught more animals, so 100 in total. It dropped to 61 the next year, so we're losing some lizards. We're 180 there, 366, 527. 2019, we're at over 700 lizards on our islands. So the experiment worked. Our populations are thriving, and so the nice thing about these very small islands is that we can catch every single individual. And every single individual, we put a pit tag in, and so you know who is whom, who survives, who dies, who has offspring, because we cut little pieces of tail, so we can do the genetics, get the genealogy, and do all kinds of other things afterwards. So for every single lizard, we measure its bite force. We flush its stomach every couple of years to get the diet, see what they're eating. We get pictures of the lizards, we measure their sprint speeds, we measure morphometrics, we get genetic samples, and so we try to measure as many things as we can do with having the least amount of impact on our populations, because these are very small populations. So what do we find? And this is completely unexpected, is that from 2014, 2018, for males and for females, this is bite force. Lizards have more than doubled their bite force in 1, 2, 3, 4 years. 4 years, 2019 is right there. And so we see enormous phenotypic changes. Like these are doubling their bite forces. This is not nothing. And all the changes happening in the first 3 to 4 years. So these changes are extremely rapid. Yeah? Very different what I would think of if you think about these kind of elements. If I would show Louis these 2 lizards from the new introduced islands versus the source population, he would tell me based on morphology, it's a different species. They're that different. And all of this change is happening very rapidly initially, and then it seems to be plateauing. Of course, this is a very dynamic system, so we really need to follow this up. So the idea is to follow these populations for many, many, many years to come and actually see how this evolves further. It's not just the absolute bite force and distribution of bite force that's changing. If you look here, for example, at the relationship between bite force and snout vent length, you can see that the slopes of these relationships stay more or less the same for males. It's the intercept that's changing. For the females, the slopes are also changing. So the females are biting much harder, much sooner as they are growing. suggesting there is a selective advantage for females to be biting hard very quickly. We can speculate for why this may be the case, and if we then plot our control island, Alico here, you can see the real difference that we have after these different years. So it's a quite spectacular change. What else do we see changing? These are the number of bite scars. So when these lizards bite each other, we can see, because there's scars that are left on the bodies. 2015 here, this is our control population from Alico that we have. And so you can see that the number of bite scars have doubled. Some of these individuals have up to 30, 40, 50 scars on their body. These things are fighting all the time. These are very small islands. We get very high densities, 200 lizards in a very small space. And so they're interacting a lot. They're biting a lot. And because they're biting a lot, you can think that there is selection, for example, for bite force. And so this may be one of the reasons why these things are increasing in bite force so rapidly on these islands. You can ask yourself as well, so what is determining survival? So we introduced 100 islands and 100 lizards on the island, 61 survived to the next year. So what determines that initial survival from year 1 to the next year? Because we have two different dynamics. So there's one, the initial survival, and then there's a subsequent survival, and we can look at what's going on. And so the survival from 2014 to 2015 is determined completely by the number of jumps. And so if you can play the video, so this is an experiment we did to measure sprint speeds, for example, and you can see the lizards here hopping from rock to rock. And so lizards that hop more survived better on this island in the first year. And so you can ask yourself, why? Why would a hoppy lizard survive better? Well, if you quantify the number of prey on the islands, you can see that on these small islets, there's lots of flying prey. So being able to jump, or being a jumper by nature, may allow you to access these more flying resources and may allow you to survive initially when you get into this very poor environment where there's not a lot of food available. Yeah. And so this seems to be an important trait explaining this initial survival. We looked at many other things, for example. Here, this is body condition, scale mass index. How does it predict future body condition? This is body condition. How does it impact survival? And so you can see that for the first 2 years, body condition is a very good predictor of survival. So from 2014 to 2015 and from 2014 to 2016. Afterwards, body condition is not a good predictor of survival. So initially, when you get to these islands, after rafting across from wherever it may be, Southeast Asia to the Mascarenes, for example, your body condition is going to be the single most important trait, potentially, that allows you to survive the initial arrival on the island. Body condition is also going to impact later on survival from one year to the next. If in 2016 you had a good body condition, you're going to have a good body condition in 2017 and 2018. But this is later on. This is a second dynamic that's being put in place in the system. What about bite force? So bite force initially here does not explain your survival. Yeah. Later on, bite force seems to be explaining quite a bit of the survival in the system. And later on, bite force is also a very good predictor of your body condition. So if you're a better biter, you're in better condition, potentially because you have access to better resources, or you can eat more in different types of prey. You can eat harder prey, you can include marine resources potentially into your diet, and so these are all things that we're looking at. But initially, bite force is not a good predictor of survival. So when you get to the island, it doesn't really matter if you're a good biter or not. What matters if you're fat and happy and you can make it to the next year. And so you can also look at relative bite force given for a given body size, and also relative bite force is not a good predictor initially, but again, it is a very good predictor later on in life. Yeah. And so it seems like bite force, one of these traits that we looked at, is not a really good predictor for initial survival. However, if you look at bite force plasticity, this turns out to be really interesting to look at. Because the bite force plasticity is how much you increased from year 1 to year 2 in your bite force. So as the lizards grow, they're going to put in more muscle mass as they use their muscles, just like we do if we go to the gym. We're going to increase our bite forces. And so this plasticity, so guys that went from 2 Newton to 4 Newton here ended up surviving on average 2015. If you were able to increase your bite force from year 1 to year 2 more, you survived to 2016. If you increased your bite force up to 10 Newton, these are the lizards that survived to 2017. So animals that are capable to capitalize on resources when they get to these islands develop larger muscles, clearly seem to be able to survive longer in the experiment. You're going to have more offspring and going to have more of an impact on the final population, because they were able to increase their bite force. No, it's okay. So because they are capitalizing on these new resources, they are able to change the population. Because bite force is heritable and the morphology behind it is heritable, these traits are going to permeate through the population. And so we see this increase in bite force from one year to the next, and this is really escalating through our population. Until we reach a level, after 3 years, of the highest bite forces that we've observed in an inter-island study across all the islands of the Cyclades. So these things are really changing very fast in a very short time. So if I'm going to conclude a few things from this study, is that the temporal dynamics, these changes are extremely rapid. I'd previously done a study where we looked at introduced lizards that were introduced 30 years ago, and we thought this was rapid. This is way more rapid than anything we've ever seen in any population that we've looked at. So changes can be really rapid in certain conditions, and animals arriving on islands can change very rapidly given the right conditions. These changes appear partially driven by competition, the increase in bite scores. So later on, as soon as the populations start increasing in density, there's an extra selective pressure for bite force. Initially, bite force doesn't matter, your plasticity matters, and your body condition matters. Later on, it does become important. So bite force, plasticity, initial body condition determine survival. We've seen this. And the initial body condition and jumping predict survival. The other thing that I haven't really shown you the data for is, but that we see the same pattern in all 5 islands. Yeah. We have huge variance between islands in the number of lizards that are present now. Some islands have over 200 lizards. One island still has 22 lizards. But those 22 all have really high bite forces. Yeah. So the dynamics are very different on the island, but there's an enormous amount of determinism in the system. Our 5 islets, every single lizard is evolving versus the same traits, towards the same traits. Yeah. And so at least in these very small islets, the selective pressure seems to be so high that you get this determinism in their morphology. So what are we doing now? Well, we keep doing the same thing for the next however many years we can find the funding and that I live to go back to the island. We're doing vegetation samples, insect availability every 5 years. We really want to see what's the effect of the lizards also on the ecosystem, these very small, fragile ecosystems. What are these mesopredators doing there? We're doing paternity analysis, working on that, so we can calculate fitness, because we know every single lizard. We know every single lizard, the number of offspring he has, how long he lived. We can calculate inclusive fitness for every single individual and see whether that's related to the traits that we're looking at. There's very, 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 very few studies that have been able to do this, and so this is a quite unique opportunity. And then finally, what we want to do, and what we're doing also, is sequencing some genomes, sequencing some methylomes, and look at what are the drivers of that variation that we're seeing. Is it pure plasticity? Is it epigenetic mediated changes that we're seeing? Is it encrusted in the genome? And how fast do these traits, are these traits integrated into the genome over time? With this, I really want to thank all the collaborators that work on this study, and I would like to thank you for your attention. If there's any questions, I'd be happy to answer some questions.
One of the key things for you is you've got a blank slate island, yeah? But why these blank slate islands in the first place?
That was the condition upon which we were allowed to introduce lizards on islands.
But there's something else in there, because clearly the lizards don't naturally want to be there, yeah?
Well, the lizards are not naturally there, probably because there's lots of stochasticity. These populations are going to remain small, and at some point they're going to go extinct by natural processes. And so the reason there's no lizards on these very, very tiny islands is because there's just too much stochasticity, and the islands are too small for the lizards to survive long term. Yeah, so we're really talking about like this size islands.
Very interesting talk. I was wondering if these islet populations, are they now comparable phenotypically to the small islet populations that are native on other islets?
Yeah, so so in 3 years' time, they have the exact same phenotype and the same bite forces as those tiniest islands that we can find in the whole Cyclades archipelago. So that change happens super fast.
Very interesting talk, Tony, and nice result as well. I would like to know if you have thought about doing some behavioral tests on these populations, perhaps something like personality, because it looks like a really good system to test many hypotheses.
Yeah, exactly. So this is one of the things we want to do. So we've done some experiments looking at thermal ecology. We find changes in the thermal ecology between islands. So the thermal environment is different, and the responses of the lizards to the thermal environment are different. But the other thing we want to really do is also do common garden experiments and do behavioral tests. Quantify the personalities, really stay lizards on the island, see who survives, who doesn't. And so there's a million things we can do on this system. So if anybody's interested in doing something on the system, just let me know. I'd be happy to collaborate.
Well, you show under some results that there's a clear increase in body size and snout length, as well as an increase in bite force. But on the bite force, you see a plateau, and you don't see it on the snout length. So what are the main cause for this plateau? Do you know if there is a reason?
Yeah, so it seems like snout length is keeping increasing. Bite force is definitely plateauing. There may be several reasons why this is the case. One of the hypotheses is that at some point you're having so much muscle in your head, if you increase the bite force, you would break your own jaws. And so this is something we're trying to test on another model system in Croatia, where we're doing biomechanical models to see how far can you push the system until it becomes limited by its own bite force, for example. So we think that's one of the reasons why this plateaus at some point.
Okay, thanks, Anthony. We will now, yes, go to the last speaker of the day, Aaron Bauer from University of Villanova, talking about New Caledonia.
Thank you. So I want to talk to you about something totally different. We're going to move halfway around the world to the South Pacific. And rather than present some sort of coherent research program like Anthony, I will be sort of giving you a bit of an update for some new discoveries, some already published, some not yet published, from reptiles, lizards in New Caledonia, to try to convince you that this is not only an interesting system, but a system that we're still learning lots of new things about. So probably most of you know a little something about New Caledonia. If you don't, it's a reasonably big island, over 18,000 square kilometers. It's about 1,200 plus kilometers off the coast of northern Queensland. And in comparison, for example, to Réunion, the human population is only about a third as much. So it's a fairly large island with a lot of diversity. So the terrestrial herpetofauna is primarily lizards. We'll skip the few snakes that are there for this talk. And those lizards come in three varieties: skinks, diplodactylid geckos, and geckonid geckos. We're going to ignore the geckonid geckos because there are only 6 of those in 5 different genera, and none of them are endemic. On the other hand, if we look at the other groups of lizards, in skinks, we've got 65 described species in 20 different genera, at least 2 additional undescribed species. 97% of those are endemic to New Caledonia. Diplodactylid geckos, 36 species described, 34 undescribed, but will be described by the end of this year. And they're 100% endemic to New Caledonia. So the levels of endemism in New Caledonia are really spectacular. And this really should be no surprise. Those of you who maybe know about the botany of New Caledonia or many other groups of animals in New Caledonia will know that levels of endemism are high for almost all groups. Just to put that into some perspective, including those soon-to-be described species, we have 149 species in New Caledonia. 140 of those are endemic. And if we look on a per unit area relationship, New Caledonia has about 25 times the species richness of New Zealand, which is shown here in comparison to some other places. Lizards are the dominant land vertebrates in New Caledonia. There's a lot of microendemism, as you might expect, with so many species packed into this island. Lizards are ecologically important for a variety of different reasons, and I just want to point this out at the end. In 1984, before my co-author Ross Sadler and I really began working in earnest in New Caledonia, there were 39 species recognized. So it's an increase of 110 species, and the point of that is really a conservation point. The point is, if you don't allow research, if you don't allow people to collect, if you don't allow people to work on these animals, what you know about your animals will be detrimental, because all of your conservation units will be totally unrealistic units. In this case, you would have thought that there'd be relatively widely distributed species, and in fact, almost all the species in New Caledonia are really very localized endemics. So, what I'm going to talk to you about today is these new discoveries in New Caledonia, and I'm going to start with what I think is the most spectacular one of these. And this is a new species of gecko that we found a few years ago. And if you look at this, it may look like just another gecko to you, but it's another giant gecko. New Caledonia is known for its giant geckos. It has the largest living gecko species in the world. It also has the second largest living species of gecko in the world, and something like the 5th and the 7th and the 9th as well. This is another giant gecko, and it's one that is not something that we identified by sequencing it and finding out that this was a cryptic species. It's an entirely new, novel form. And here's a CT of this animal, and just to point out a few things of it, this is the holotype of the animal. The holotype's 150 millimeters in snout vent length. So if you're used to varanids or other big lizards, this isn't really big. But for a gecko, this is huge. But if you look at this animal, you can see things like the epiphyses in the legs and in the feet are not fused. And the same is true in some of the joints in the skull as well. So this animal's 150 millimeters snout vent length, and it's not grown yet. It's still on its way. We have seen an individual as large as 180 millimeters, but suspect that they will grow to 200 or more, which would put them up in the very, very top of the largest geckos. It has a number of different features. I'm not going to bore people who are not into osteology about these, but the arrows point out features of the skull that are unique to this form, which made it a little difficult to decide where does this go. Does it fall into one of the genera of existing giant geckos or not? So where is this animal from? Just to orient you on New Caledonia, there's the Grande Terre of New Caledonia with the Loyalty Islands on this side, the Ile de Pin down here, and then Walpole Island, which we'll talk about a little bit later, down here. This is the area where the animal is found, and this is a blow-up on the map. It's right on the border between the Province Nord and the Province Sud of New Caledonia. It's a steep forested area in the Chaîne Centrale. And a little bit, and it is a very little bit that we know about the ecology of this animal from two different sources, dietary analysis of the holotype and then of a second specimen. We know that they eat a minimum of 14 taxa of arthropods. You can see some of the different types that are there. One thing I want to point out is amongst the coleopterans that are eaten include some aquatic or semi-aquatic taxa. Also, they eat fruit of a variety of sorts. Here is a plant part that's inside this particular animal, inside the holotype. So they have a fairly diverse diet. Now, that a potentially aquatic beetle is interesting because all of the individuals have been found active at night on stones adjacent to streams or in streams. If you don't know geckos, that may not mean much, but if you know geckos, there aren't aquatic geckos. And this is basically at least a semi-aquatic gecko. And one individual that is not part of the preserved specimens we have actually swam to escape capture. So this animal has a striking resemblance to the genus Dactylocnemis in New Zealand. So here's our new thing from New Caledonia. Here's Dactylocnemis from New Zealand, from northern New Zealand. Another interesting thing is that this sort of aquatic or semi-aquatic behavior is only known in other geckos in Dactylocnemis and in 2 of the 280 species of Cerdidactylus in Southeast Asia. And the fact that this animal is associated with streams is the reason why we didn't find it until now. It turns out that Ross Sadler and I walked up this stream bed in 1992 and collected and never saw this thing, because who's going to be looking for geckos on wet rocks in the middle of a stream at night? So the unexpected ecology of this animal has sort of kept it hidden for this time. So morphologically, it has some similarities with this animal, which is the largest gecko that ever lived, known from a single specimen, which we thought until recently came from New Zealand. There were no data with the specimen. But now we've sequenced the ancient DNA. The specimen is about 200 years old. And it suggests that this animal is from New Caledonia, not from New Zealand. So the question is, for this new gecko, is it related to that gecko, that giant gecko? Is it related to Dactylocnemis in New Zealand that it looks so much like? Or is it related to some of the living giant geckos that we've known for years that occur in New Caledonia? And just if you take a look at something that's sort of easy to wrap your head around, if you look at the feet of these animals, you can see here's the new species up here. Here is a Hoplodactylus, or related to Dactylocnemis in New Zealand. And here is Nyaro gecko, which is one of the giant geckos in New Caledonia. And you might sort of look at this and say, well, this looks a bit more like this than it does like this. The digits are slender, they have this long distal portion here. So maybe that hints that they are in fact more closely related to the New Zealand geckos than they are to other giants in New Caledonia. Nope. It turns out that the giant gecko from 200 years ago, that huge gecko, falls in this group, which includes all of the giant geckos living in New Caledonia today, plus some other smaller bodied geckos. Here's the New Zealand geckos up here, and our new species falls in this blue clade, which is a clade that includes Dairogecko, which is the smallest bodied group of geckos in New Caledonia, typically in the 35 to 42 millimeter snout vent length range. So this implies that there's probably been at least 2 different origins of gigantic size amongst geckos in New Caledonia. So I want to switch now from that giant to another giant, talk about giant skinks, and this is work largely done through Hervé Jourdain. I think Hervé is here someplace, I think. There he is. At ERD in Noumea. And this has to do with the genus Phoboscincus, which is the largest skink in New Caledonia. And this is an interesting animal. It occurs, or at least the specimens that we know of, are only from this Ile des Pins area. Here's the Ile des Pins here. And there's a bunch of little satellite islands around here, and we'll come back to that in just a second. So Phoboscincus bocourti was known from only the unique type for over 125 years. It was rediscovered. It was rediscovered on Ile Bros, which is a small island off the coast, the west coast. But, and with the material from that rediscovery, we were able to show that bocourti is related to its supposed sister taxa, Phoboscincus garnieri. So the 2 giant geckos in New Caledonia are in fact each other's sister group. But Hervé was able to find a population on another island, on Île Bayonnaise, and was able to collect some preliminary data on these animals, and I think further data is forthcoming. So just to give you an idea about what a giant skink looks like in terms of size, you can sort of guess when you see these things here, they look like they're big, but when you see people's hands in here, you get a sense for the size of these skinks. So these are really large animals, and you can see here that the snout vent lengths are here well over 200 millimeters. And the island that they occur on, Bayonnaise, is a very small island. A number of different individuals were observed. We have some data, from diet, indicating that they are able to eat other skinks in this case. A camera trap recorded individual feeding on a dead ratus, which was a bait animal. It wasn't a ratus that occurred on that island. And the activity period is interesting because it goes all through the day and into the evening, which is an odd thing for a lot of skinks to do something like that. And so if we look again at the map, here's the Ile de Pin here, down here is Brosse, and Bayonnaise is up here. So there's a possibility that it may also occur on some of these other western satellite islands of the Ile de Pin. And this is a very touristic area, beautiful area that lots of people go to, but there's still a lot of things that we don't know, like people are not going onto some of these islands at night. People are not really looking very much in the forests, for example, that occur on the main island of the Ile de Pin. There needs to be more work done there. The next new discovery I want to talk about is not so spectacular in terms of size, but it's biogeographically interesting because it comes from Walpole Island. And Walpole sits down here. Walpole is known herpetologically because it had myelinaeid tortoises on it. Walpole is quite small. If you look here, it's about 2 square kilometers. It has pandanus and remnant dry forest on it. And the only other reptiles that have ever been recorded are Lepidodactylus lugubrus and Amoya cyanura, which are sort of pan-Pacific species. And this is what Walpole looks like. There's the west coast with more sort of formidable cliffs and the more accessible east coast. And here's the habitat. There's Amoya cyanura up here. And then here is the new species of skink from Walpole, which is Epibeta insularis. And it's got a close relative on the mainland and elsewhere in New Caledonia, which is Epibeta nigrofasciolatus. But there are some differences that I think you can easily see. Darker sort of melanistic coloration dorsally, a duller ventral surface, whereas here you have a very bright surface. And there are a few other minor scalation differences. And it turns out that this animal is not something that is related very closely to Epibeta that are in the Loyalty Islands, which are 140 kilometers away to the north, or on the Ile de Pin, which is 140 kilometers away to the west. Instead, if you look at this, these are all Epibeta nigrofasciolatum from the Ile de Pin region. This is a Loyalties animal. These are all from the Grande Terre of New Caledonia, and the sister to all of these is this new species, Epibeta insularis, with a 5 to 14% genetic difference in D2 between these and the other forms. So this is sort of interesting. It's the first endemic from Walpole. Walpole is very isolated. And the fact that it's sister to the rest of the entire New Caledonian region is also interesting. So those are the discoveries I wanted to highlight, but there's still a lot of other things going on in New Caledonia as well. This is a blow-up of the Ile de Pin, and we recently described another new genus and species from here. This is Cunisaurus albiorus. This is its habitat from this part of the Ile de Pin. And the Ile de Pin, as I said, is a very touristic area, but a lot of the forested regions remain undersampled, and more information is really needed from there, and new discoveries might be found. To give you an idea, just concentrate on those yellow bars. It doesn't even matter what they are. Those are all taxa that are strictly endemic to the Ile de Pin or its satellite islands. So within this New Caledonian sort of hotspot with huge endemism, we have this smaller area, which itself has a high level of endemism within the area as a whole. In addition to the Ile de Pin in the south, if we go to the far north, we get to the Beleps up here. And in the Beleps, in 2012, we described 2 new giant geckos from that area. One endemic to these islands, one on these islands and also extending onto the northern mainland. And Ile Baba here has an endemic species of Diuro gecko. This is one of these little geckos related to our new giant form. And then finally, the Loyalty Islands, running here along the east coast of New Caledonia. The Loyalties also have 4 endemics, including 1 snake here, Rhamphotyphlops wiley. But the lizards are forms that, except for Emoia loyaltiensis, we didn't recognize as being endemic before. These were ones that are, in fact, cryptic taxa that we were able to resolve using a combination of morphology and sequencing data. So just to sort of summarize, despite having spent 40 years essentially between Ross and I working in New Caledonia, there's been, when we've had during this period a 3.5 times increase in the number of recognized taxa, new forms still are being discovered, and these include not only things that are cryptic taxa, but things that are true novelties that are morphologically easily distinguishable, but for example, like this new giant gecko, occurred in a habitat that we never thought to look, or was doing something that we didn't know it did, or like the phoboscincus, which is sort of a rediscovery, has an activity period that may have precluded us from finding it or others from finding it before as well. The discovery of that giant gecko, as I said, suggests that we have multiple derivations of giant geckos in New Caledonia. The unique ecology of both that gecko and the phoboscincus, as I said, are things that probably kept them concealed for this time period. And then I'd like to end by sort of emphasizing the satellite islands of New Caledonia. In the past, I think we traditionally thought that New Caledonia was a place where the diversity was on the Grande Terre. The diversity was all on this main old high island, when in fact, when we look at these lower islands, in some cases coral atolls, in other cases pieces of the Grande Terre that are now isolated, we find that these also have their own endemics associated with them. So we really have these four areas of endemism outside of the Grande Terre. We have the Ile de Pin and its satellite islands. We've got the Beleps and associate islands in the far north. We've got the Loyalty Islands, and we've got now Walpole for the first time in the far southeast as being a representative of a place that also has an endemic. So with that, I'll end and just sort of point out again that New Caledonia remains, after all this time, really a major hotspot with a huge amount of diversity. But where we stand right now, we're reaching probably an asymptote in terms of what we know about the systematics of the lizards and the few snakes that are there, but we know almost nothing about the ecology of any of the animals that are there. And so this is, I think we will move into a new era in herpetology in New Caledonia when we get more people who are in situ in New Caledonia, who are able to participate in looking at these sorts of things, spending time in one place through the seasons to follow what's going on with a lot of these organisms. So I'll just conclude by thanking a variety of people, not the least is Hervé, who is sitting out there at IRD, who's been sort of a leading force, despite being an ant person, is a leading force in terrestrial studies of all kinds in New Caledonia, has been very helpful over the years in reptile studies. Matthias, who is up there, who is sort of one of the shining stars of the future for working in New Caledonia, and John down here in the corner, who is one of my students from the States, holding the holotype of that new giant gecko. And I'd just like to thank the National Science Foundation of the US and my university for funding all of this, getting me around the world, and thank Crystal for inviting me to participate in this symposium, and of course the organizers for this symposium and the meeting as a whole, which has been a wonderful experience. Thanks.
Okay. Thank you, Aaron. That was so interesting. Are there any questions? I know that the poster session starts soon, but I suppose if you want to go to the posters, please go, and if there's questions, please hang around. Questions for Aaron?
Yeah, Søren Fauby, Gothenburg University. perhaps cheating a bit, because I think this question is actually to perhaps all of you. I have to start by thanking for a really, really interesting symposium, but I was wondering if some of these patterns are driven by human-driven extinctions. Crystal started by talking about that in the islands we are on now, there are, I think you said more than 10 extinct reptiles, and there is about 50 or so known extinctions in total. So that must mean that there's a shitload of unknown extinctions. So I was wondering if how many of these patterns could actually be. So anthropogenic, when you in the end talk about that these things are endemic to really, really tiny islands, and you know that one of them was only found on islands without rats, or when the second talk was discussing that there is a drastic difference between the fauna in Sao Tome and Principe, if the part of that is just driven by local extinctions rather than independent colonizations?
Yeah, I mean, I think that's an excellent question, and certainly some of these things, like Phoboscincus bocourti was certainly on the mainland. It was certainly at least on the Ile de Pin, even though it's only on the satellite islands now, or that we know it from the satellite islands. And that's certainly true for, in New Caledonia, that this would be true for many things. And then we have the real wrench in the works, which is when you look at fossils. And from New Caledonia, we have a wonderful fossil locality at Pindai, which we have material that goes back before human arrival in New Caledonia. And we have new genera. We have a new genus of diplodactylid gecko we only know from those fossils. There are varanid lizards that we only know them. We're not quite sure whether they precede humans or they came with humans. But you know, looking at that is very sobering for me, because I can put this all together and I have a nice idea about what the patterns are, and then I look at what little evidence we have of what was once there, and everything you thought you knew goes into the garbage. So yeah, I mean, I think we always have to keep in the back of our minds that we are kidding ourselves if we think that the patterns we see today are necessarily reflective of the pre-human situation.
More questions? For anyone? Well, what about if anyone is going over to the poster session, you can go and you can individually grab these guys and ask your questions, or just talk about collaborations and that sort of thing. Because I have to say, I was pretty blown away by these talks. I don't know about you guys, but I had no idea what had been lined up. Well, that's not true. I had some idea, but wow, in the end, I have all kinds of questions for them, ideas, and things are going through my mind. So, hello. So please, I'm sure you must have the same. So let's go over to the posters and get your beer there and talk some more, right? Thank you. Thanks to everyone for coming. Oh, and thanks to the speakers.